The overall goal of this two-step laser writing procedure is to obtain elastomer-based micrometer-sized light actuating structures.
The method can help to answer key questions in the fields of microrobotics and microfluidics, such as how to create a single microstructure with complex movement.
The main advantage of this technique is that the molecular orientation can be manually patterned in the microscale. Thus, the specific form of the actuation can be arbitrarily controlled.
So this method can provide insight microrobotics, but it also can be applied to different systems like tuning in photonic devices or material responses to different stimuli. Demonstrating part of the procedure will be Daniele Martella, a postdoc from my laboratory.
To begin, clean a 3 cm diameter glass cover slide with acetone and lens tissue. Then, using forceps or tweezers, place some glass microspheres on the slide about a ½ centimeter from the center point. These function as spacers. Place a 1 cm diameter microscope slide on the spacers and gently press it into position. Next, add about 2 microliters of UV curing glue into the gap between the slides at the 3 different points where the spacers are located. Before the glue penetrates too far into the gap, use UV light to cure the glue. This forms the cell for the experiment. Now, along the edge of the cell, drop about 10 microliters of IPL resin. Wait for a few minutes until the liquid is infiltrated into the